es el último trabajo que publicamos recientemente a principios de año, que habla sobre la interacción entre las células T, la mielina en el sistema nervioso central y el envejecimiento. Si no os importa, para la presentación cambio a inglés, porque si no acabo inventándome palabras que no existen. Después de 12 años en Reino Unido, todavía estoy mejorando el español científico, que digamos. Pero luego las preguntas, si queréis, se pueden hacer tanto en inglés como en castellano, me da igual. So let's begin. As Diego has said, my group and my research career has focused on understanding myelin and how myelin is formed and restored, especially in the central nervous system. As you all know, myelin is a lipid-rich multicentric layer that is formed surrounding the axons, both in the peripheral and in the central nervous system. In the peripheral nervous system is formed by Schwann cells, while in the central nervous system is formed by oligodendrocytes. Oligodendrocytes are a polydendritic-like cell that extends processes and can engage up to between 50 and 100 axons in the central nervous system, and extends their membrane around these axons and gives several wraps around the axons in order to form the myelin, which basically is essential to control circuits. And obviously the main function of myelin is to give axons trophic support and maintain fast saltatory conduction of action potentials. In the central nervous system, oligodendrocytes arise from a population of brain stem cells known as oligodendrocyte progenitor cells, or OPCs. OPCs account for about 5 to 8% of the central nervous system cells, even in adulthood, and they are basically distributed throughout the whole central nervous system. They are responsible for giving rise to oligodendrocytes both during development and in adulthood during basically in response to different experience, but also especially in response to damage in the central nervous system. Although myelin has been mainly studied in the context of primary demyelinating diseases like multiple sclerosis, which is going to be the focus of this talk later on, now is well accepted that we can see myelin alterations as a hallmark across different neuropathologies. So just said, we can see them in multiple sclerosis, which is the primary demyelinating disease, or adrenoleukodystrophies, which represent a genetic disorder that leads to demyelination. But we also see myelin alteration in other psychiatric disorders, such as autism spectrum disorders, schizophrenia, or other neurodegenerative diseases, including Alzheimer's disease or ALS or Huntington's. For the purpose of this talk, I will center my focus on multiple sclerosis as the prime example of a demyelinating disease where we have an interplay between inflammation and myelin repair. As you all might be aware of, multiple sclerosis is the most common neurodegenerative disease among young adults, affects nearly 3 million people worldwide, and is usually diagnosed between the age 20 and 40. And 75% of the patients with MS are women. It has quite a strong gender bias compared to other diseases. It's also known as the disease of the thousand faces because in multiple sclerosis, or MS, the immune system recognizes myelin as a foreign body, and depending on the area of the CNS that is altered, you can have a wide range of symptoms like the one represented in this diagram, which for years have basically made very difficult to give a thorough diagnosis of the disease at the beginning of the symptoms, and obviously this is quite relevant later on for disease progression. Most patients present this type of disease course, if you want to call. So most patients have a relapsing remitting MS, especially at the beginning when the disease is diagnosed. In this relapsing remitting MS, what happens is usually the immune system targets myelin. As I said, it recognizes myelin as a foreign body, and that leads to an accumulation of disability. This disability can have a wide range of symptoms from like spasms, sleepiness, not being able to talk, depending on the area of the CNS that is affected. But usually, especially at young ages, these relapses calm down and go back nearly to full recovery, though slowly with each relapse, we keep accumulating disability over time. Most of the patients progress with time to what we call secondary progressive MS. And in this case, there's no relapse in remitting phases. We basically just turn to a more neurodegenerative phase of the disease in which patients just accumulate progressive disability, and this doesn't restore, or they don't never get back to functional recovery. And about 10 to 15% of the patients, they basically accumulate progressive disability with no recovery at all from the beginning of the disease. This progressive disability is obviously associated with brain atrophy and also accumulative axonal loss. But if we look back to the biology of how this happened and where these relapsing remitting phases link in the CNS, what we see is the following kind of very simplified diagram. Here you just have a schematic view of the central nervous system where you have like neurons in gray and oligodendrocytes forming myelin around the axons in yellow. And here on the left side, you have like a transversal view of these axons, similar to whether if we make a cut here and look at the axons kind of from the screen towards you. So what you can see here in white are the axons transversally cut, and in blue you see the developmental myelin, the myelin. This is from a rodent model, the myelin that is formed during development. As I mentioned in MS, T cells and the immune system recognize myelin as a foreign body. They target myelin and oligodendrocyte died, leaving the axon demyelinated. So you can see here on the left, and this obviously leaves the axon vulnerable to subsequent neurodegeneration. Unlike neurons in the CNS, which are refractory to regeneration, myelin has a remarkable regenerative capacity. The pool of OPCs that I mentioned before, these oligodendrocyte progenitor cells, which represent an unmatched niche of stem cells for repair, react or recognize this myelin damage, and they differentiate into myelin-forming oligodendrocytes, leading to functional recovery, with one slight difference compared to developmental myelination. The myelin that is formed in adulthood is slightly thinner, as you can see here in this transversal picture. So basically, if we want to make a very simplistic diagram, what we have in the early stages of MS is a very tight balance between inflammation and regeneration. There's an inflammatory attack that coincides with a relapse in patients that eventually shuts down because inflammation goes down. And at the same time, we get a repair phase in which the lost myelin is restored. And this more or less keeps functional recovery, at least to a certain extent. However, when disease progresses, and especially with aging, which I will talk later in a bit more detail, this balance is completely tilted. Inflammation outweighs myelin repair. There's no myelin regeneration in this context, and that translates into progressive axonal loss, neurodegeneration, and obviously the accumulation of irreversible cognitive and motor disabilities in patients. If we look at the treatment landscape that we currently have in MS, it's one of the few successes between brackets for neurodegenerative diseases. Since 1990s, we have had a number of treatments that have managed to control the disease to a certain extent, but all these treatments are effective mostly in the relapse-remitting phase because they all control the attacks that the immune system has in myelin. And a couple of them have recently been approved for the progressive phases of disease, but they do not stop disease. None of them has the ability to stop disease. They only kind of slow down progression by a very mild percentage, something like 10 to 20%.
Tengo una preguntita. El tema de la...
Se cortó a mitad de la pregunta.
¿Ahora sí?
Ahora sí.
¿Hay relapsing remitting en la neuritis óptica asociada a esclerosis múltiple o ahí es solamente... ¿Qué pasa con la visión, digamos, en la relapsing remitting?
Ahí es un poco más complejo porque hasta ahora se consideraban la misma enfermedad y ahora la neuritis óptica se ha separado como clasificación en la enfermedad de la esclerosis múltiple. En ambos hay una relapse y remission, lo que pasa es que depende de la dureza del ataque en el caso de la neuritis óptica se puede perder más o menos visión y a veces no se recupera del todo. No te puedo decir exactamente porque no he trabajado tanto en neuritis óptica si los periodos de relapse y remitting son muy parecidos a esclerosis múltiple, pero sí que sí que es cierto que los pacientes, muchos de los primeros relapses son visuales. Pero creo que también tiene que ver más con el hecho de que el paciente eso lo nota.
Claro.
Entonces lo reporta mucho más al neurólogo, mientras que, por ejemplo, cosas como que se nos duerman las manos, que también es un síntoma de uno de los ataques en pacientes de esclerosis múltiple, muchos de ellos lo ignoran porque es algo que nos pasa a todos y no le damos la mayor importancia. Pero no sabría decirte exactamente.
Yo sí trabajo en neuritis óptica, pero en un modelo de neuritis óptica primaria, y no tengo al día de la literatura de si hay remisiones en el nervio óptico. Eso no lo vi todavía escrito. Así que...
No sabría decirte con seguridad y no me atrevo por no dar una información equivocada, no me atrevo a decírtelo. Sí que es cierto que cada vez hay como más, intentan distanciar más ambas enfermedades y parece que el patrón no es el mismo.
Exacto.
Pero no sabría decirte si tienen relapses remitting, la verdad.
Muchísimas gracias.
Nada, gracias a ti. So as I said, there's no therapies actually that really slow or stop the disease. And currently we have no therapies that will enhance myelin repair, which is an ultimate, the most potent neuroprotective mechanisms that we have to protect axonal health and prevent neurodegeneration. Therefore, my group and others, what we have been focusing is on trying to understand how remyelination happens and how we can boost this process. Remyelination, like any other regenerative process, is obviously a very complex, tightly regulated, multi-step process. Here, I'm going to give you a very simplistic summary of and a step-by-step process in remyelination, or at least to highlight the main processes that happen in CNS remyelination. So here you just have another schematic view of the normal white matter. Axons are in blue, oligodendrocytes in yellow, and then we have microglia, astrocytes, and the pool of progenitor cells, or OPCs, which in adulthood are mostly quiescent. When the immune system targets myelin, oligodendrocytes die, demyelination happens, and myelin debris release, which is inhibitory for the regenerative process, and therefore it has to be removed. So microglia and astrocytes kind of mop up or remove myelin from the area of damage, and they secrete cytokines and chemokines that lead to the activation of the quiescent pool of OPCs, which get recruited to the area of damage, where they proliferate, and eventually they differentiate into myelin-forming oligodendrocytes and restore lost myelin quite efficiently in the early stages of the disease. However, as disease progresses, this process is impaired, and we have identified between different groups 2 main bottlenecks, which are progenitor recruitment and progenitor differentiation. There's different types of patients, and we know that in some of them, they get enough OPCs to the lesion, but these OPCs cannot differentiate. And in some others, they don't get enough OPCs to the lesion. So both processes appear to be impaired in MS patients. Hence, what many research groups are doing, including us here in Alicante, is to understand and identify molecular and cellular mechanisms that might regulate this process. So in my research group, what we are trying to do is to identify intrinsic factors of the environment, and especially also systemic factors that might modulate this process in order to enhance the ability of the pool of endogenous progenitor cell of OPCs to form new myelin and repair the lost myelin in MS. For the sake of the talk today, I'm going to focus on 2 systemic factors, which are aging and the immune system, and I'm going to talk a bit about the interplay of these 2 systems with CNS remyelination. So as you all have heard probably sometime, the CNS has for long been considered an immune privileged organ. For many years, we have thought that the immune cells do not get into the CNS unless it's in the context of injury. However, since I would say 6 years ago, this dogma has been challenged, and we now know that there's cells from the immune system in the healthy CNS. We can find CD4 T cells or CD8 T cells in the healthy CNS parenchyma early after development, and also during adulthood, albeit in very low numbers. However, even if they are very few of them, they seem to be crucial for processes like learning and memory or synaptic pruning. Thus, there's a very tightly regulated interplay between the immune system and the CNS. Additionally, work done by many groups in other organs have shown that T cells are key for regeneration in other organs. Here we just have an example of the gut, where T cells have a very closely regulated crosstalk with stem cells from the gut, but they have been shown to regulate stem cell behavior in the muscle, hair follicle, in the skin, and so on. So if we put these 2 concepts together, we could also speculate that T cells might have a role in CNS remyelination. And this was shown already about 20 years ago, but this seminal manuscript published by Beaver et al., in which they use a model of toxin-induced demyelination, which is called the lysolecithin-induced demyelination model. And it's a model that I will mention several times throughout the talk, so I'll just give you a brief explanation. So what we do in this model is you inject a toxin known as lysolecithin in the ventral white matter of the spinal cord here, and basically the toxin is is tropic for the lipids, and therefore it targets lipid-rich cells like the oligodendrocytes, kills the oligodendrocytes, and triggers quite a fast a focal lesion here. It doesn't expand across this; it's very focal, and that leads to a remyelinating response that is very well characterized and has very specific time points for different steps. And one of the advantages of this model for this topic is that the damage is not immune mediated. It's totally independent of the immune system, and therefore it allows to study the role of the immune system, like T cells, in the repair or remyelinating phase. So here what you have in the left is 4 electron diagrams where you see the lesion. You can see the normal developmental axons, which are thicker in the bottom part of the pictures. And then in the upper part is the area that was demyelinated. And you can see that there's less myelin, and the myelin that is there is thinner. So what they observe here is that in the wild type, they observe quite a robust remyelination, but this was significantly impaired in all the 3 mutants in which they manipulated the immune system. Here they have RAG1 knockouts that doesn't have T or B cells, a mutant for CD4 T cells, or a C57 black 6 mice in which they depleted CD4 T cells with an antibody. And they saw that in all of them, remyelination was impaired. Therefore, T cells are not only the bad guys, between brackets, in MS, but they are also required for an efficient CNS remyelination. However, I'm sure that you all remember your immunology textbooks from the university, and there's a wide range of CD4 T cell subsets, and each of them have a different function depending on the transcription factor and cytokines that they express. And this is also the case for remyelination. Not all CD4 T cells have the same role. We know now that, for example, Th17 cells are negative regulators. They impair CNS remyelination, while regulatory T cells might have a positive role in this process. So today I'm going to focus specifically on regulatory T cells because they are key as anti-inflammatory, but especially they have very relevant regenerative properties in the CNS. And the reason why I focus on this was before I joined my previous lab in Belfast, they just published this paper in Nature Neuroscience where they describe for the first time that regulatory T cells do actually have regenerative properties and they can enhance myelin regeneration in the CNS. They can secrete factors that enhances the differentiation of OPCs into myelin-forming oligodendrocytes in vitro, as you can see here by the expression of MVP or myelin basic protein. And when they depleted mice from their endogenous Tregs, using a transgenic mice that I will explain later on, they observed that remyelination was also significantly impaired in this lysolecithin-induced demyelinating model, suggesting that out of all the CD4 T cells, regulatory T cells seem to be key in this regenerative process. So here I have introduced the first kind of candidate of the interplay, which is from the immune system. We are going to focus today on regulatory T cells. And the second systemic factor I wanted to mention in this talk is aging. Aging is the major risk factor in MS, and it is now well established that the age of onset of the disease, but all the age of the patients are the key factors that determine when a patient switches from EDSS, acquires EDS6, which is one of the major disability scores. And this is mainly because with age, like any other regenerative process, CNS remyelination is significantly impaired. Here you just have 2 electron micrographs from a toxin-induced demyelinating model in a rodent at the same time point, 28 days after the lesion is formed. And you can see here that while the young rodent is highly efficient at remyelinating, you see all these electron-dense circles around the axons. The aged rodent is pretty impaired, and there's virtually no remyelination at exactly the same time point. And this is mainly happening because the aged CNS is refractory for myelin regeneration. There's many steps that fail in the aged CNS. On the one hand, OPCs are not able to respond to pro-differentiation cues. Here you just have a young OPCs in vitro, and these are aged OPCs, and you can see that the aged ones do not express myelin proteins like CMP or MVP. They have an impaired differentiation. Macroglia and macrophages have an impaired phagocytosis. They are not so good at mopping and removing the myelin debris. Therefore, myelin debris stays for longer in the lesion, and this impairs remyelination. And at the same time, a lot of physical properties of the CNS changes. Here you just have the stiffness as an example, and all of these regulate remyelination. Hence, considering these 2 factors, that regulatory T cells are regenerative and they are needed for adequate remyelination, and that remyelination is impaired with aging, we wanted to know what was the interplay or the role of these regulatory T cells with aging. So one can hypothesize that if regulatory T cells are needed for remyelination and remyelination is impaired with aging, we might have less Tregs or regulatory T cells as we age. It is now well described in the literature that it's actually the contrary. When we look at the immune organs like the spleen or lymph nodes, we do have more regulatory T cells as we age. So we first wonder whether this was also the case in the CNS. Maybe the CNS is slightly different and we don't have more Tregs, but the same thing happens in the spinal cord and the brain. We get more regulatory T cells in the CNS, and we also get more tissue resident regulatory T cells as we age. So this was a bit puzzling, and we were a bit, questioning this. So if we have more regulatory T cells and they are meant to increase remyelination, why does remyelination still fail with aging? So the thing we wanted to address in this project was, how does aging affect Treg myelin regenerative capacity in the CNS? And how is it affected? And if so, how can we manipulate this impairment in order to restore remyelination in the CNS? So before starting the project, we went back to the literature to see what was known about the regenerative function of Tregs and aging. And to be fair, these regenerative functions were first described about 10 years ago, so they are quite new compared to other functions of Tregs, like their anti-inflammatory properties. And very little was known in the context of aging when we started this project. We only found this manuscript published by Diane Mathis from Harvard, in which they studied something similar, but in muscle. And what they observed was that in all mice, when we injured muscle, we get less Tregs into the injury site, and this translated into a poor regeneration of the muscle. However, when they increase the number of Tregs in the aged muscle, this regenerates virtually like a young muscle, suggesting that there's a migration problem. So we were wondering whether, even if we have more numbers of Tregs in the aged CNS, when we make a lesion, a demyelinating lesion, and we lose myelin, whether they might not be able to get into the lesion area, and that might be why remyelination is not so efficient. We did this looking at the number of Tregs in the focal lesions of two different models of demyelination. The first one is the lysolecithin model that I have mentioned before. And the second one is an immune-mediated demyelinating model known as experimental autoimmune encephalomyelitis, or EA. And to our surprise, we saw that in both cases, we virtually got the same number of T cells in the lesion, suggesting that it's not a migration problem like what was described in the muscle, but it's something else that doesn't work in the CNS and limits myelin regenerative capacity of Tregs. So we next consider that, well, aging is basically inhibitory for most processes, and it creates a very limiting environment, especially in the CNS. So we wonder whether if we just increase the number of Tregs, we might be able to overcome this problem. So we went back to immunologists, where they have a very well-established model to expand Tregs systemically. They basically give a cocktail of interleukin-2, which is a survival factor for Tregs and a proliferation factor. And with that, they basically increase the number of Tregs in the whole system, blood, spleen, and lymph nodes, for about threefold, as you can see here. And then what we did is we look at whether this would improve myelin regeneration. First, we tested this in young mice, which are highly efficient at remyelinating. So if the system worked, young mice would remyelinate better when they have more Tregs. And that's exactly what we saw in our lysolecithin model. We expanded Tregs in young mice, and then we look at the number of oligodendrocytes, which are identified here by the co-expression of OLIG2, which is a marker for the whole lineage, and also CC1, which is a marker that identifies mature oligodendrocytes, myelinating oligodendrocytes. And here you just have the lesion demarcated by the dotted line. And you can see in the graph that we have more oligodendrocytes when we have more Tregs. So hypothetically, the system is working. So next, we wanted to check whether this was also the case in aged mice. We did exactly the same model, but in this case, it was 15-month-old mice. When we expanded Tregs, we did not see that increase in oligodendrocytes in the lesion, suggesting that even if we have more Tregs, we are still not able to boost the system. Therefore, we hypothesized that there might be something intrinsically impaired in Tregs with age that limits their myelin regenerative capacity. And this is quite important because since the regenerative properties of Tregs were published, there were a number of clinical trials testing whether expanding Tregs using interleukin-2 in patients could be beneficial and improve remyelination. But what our rodent model suggests that this might be beneficial for patients in the early stages of the disease, but might not actually help patients in the later stages of the disease or more progressive phases. So therefore, we will go back to the same kind of black box in which we won't be able to boost remyelination in the later stages of the disease. So we next wanted to know whether this impairment is reversible, and if so, whether we can find a way to kind of restore the capacity of Tregs to boost myelin repair. So first, we wanted to know whether Tregs are intrinsically impaired, and to that, we went back to a very simplistic in vitro model. We took Tregs from young and aged spleen, isolated them, and put them in vitro with neonatal OPCs. The reason why we took neonatal progenitor cells or OPCs is because neonatal OPCs are programmed by default to respond to myelinating, pro-myelinating cues and become oligoandrocytes. So with that, we co-cultured OPCs alone, OPCs with young Tregs, or OPCs with aged Tregs. And then we look at the number of oligoandrocytes that were formed after 5 days in vitro, identified by the expression of 2 myelin proteins like CMPAs in gray and MVP in magenta. And as you can see here in the picture on the graph, young Tregs were highly efficient at driving differentiation of OPCs into oligoandrocytes, but aged Tregs were not able to do so, suggesting that they have some sort of intrinsic impairment. To then test whether this was still the case in a more representative ex vivo system like brain slices, where we have axons and we can see if these oligoandrocytes can wrap myelin, we repeated the same experiment, but this time with cerebellar brain slices. We incubated cerebellar brain slices with young Tregs or with aged Tregs, and then look at myelination as a readout of the number of axons that are identified by NFH in gray that are basically covered by MVP in magenta here, which represents myelin. And we saw kind of similar pattern. Young Tregs enhanced myelination, but aged Tregs were not as efficient doing so, again, suggesting an intrinsic impairment. However, we did see a difference compared to the previous experiment, and it's here there's also not a clear difference between these two, suggesting that maybe if you put the Tregs in a more complex environment where they can survive longer, and if this environment is young, in this case they were neonatal slices, this might restore their capacity. And this was quite intriguing because it might suggest that this intrinsic impairment of Tregs to enhance myelin repair might be reversible, and they might be able to be rejuvenated. So we next asked whether we can rejuvenate aged Tregs and restore their capacity to enhance myelin repair in the CNS. To do that, we moved to an in vivo experiment in which we took FOXP3-DTR mice. So for those that are not familiar with the mice, FOXP3 is a transcription factor that is a master transcription factor for regulatory T cells. And these mice express under FOXP3 promoter 2 things: GFP, which allows us to identify endogenous Tregs as GFP positive cells, and also a diphtheria toxic receptor. So we could deplete the endogenous Tregs by giving diphtheria toxin at different time points. And then once the endogenous Tregs were depleted, we could adoptively transfer and give these mice wild-type Tregs that come from either young or aged mice. These wild-type Tregs are not green, so we can distinguish the endogenous from the adoptive transfer ones. And they are also resistant to diphtheria toxin, of course, so they will not be depleted by the diphtheria toxin, and this allows us to basically substitute the endogenous population by either young or aged Tregs. And then we went back to our lysolecithin-induced demyelination. And the first thing we looked, obviously, was at the number of oligoandrocytes in the lesion, again, using OLIG2 and CC1 as a marker of oligoandrocytes. And bear with me in these like 4 groups. So basically the 2 gray ones are our controls. The light gray is the control group, normal young animal with older Tregs, and the gray one is the Treg depleted mice, which basically show the same as in the previous Nature Neuroscience paper. When you don't have Tregs, you get an impaired oligoandrocyte differentiation. This is rescued when we give back young Tregs here in light blue. But surprisingly to us, this is also rescued when we give aged Tregs, suggesting or indicating that if we put these aged Tregs in a young environment, we might be able to rescue or restore the regenerative capacity. So, I mean, we obviously look at different markers and validated this also for NFH and MVP. So then we were like, this is good news in the sense that, okay, with aging, Tregs lose their capacity to enhance CNS myelin repair, but this is reversible. However, obviously, as you can imagine, this is very difficult to carry out in patients. You cannot, transferring cells from young and aged patients is not that easy. So we needed to find something more specific that allow us to either rejuvenate Tregs or to mimic the function of young Tregs in the CNS. And to do so, we went to do some bulk RNA sequencing to try to understand what is different between young and aged Tregs. And to our surprise, and to cut a long story short, young and aged Tregs are vastly different. They have about 1,700 genes differentially expressed. There's about 1,500 genes that are highly expressed in aged Tregs, while around 300 genes are lower expressed in aged Tregs. And with this, you can probably make two hypotheses. There's two things that can happen. Either something that is inhibiting CNS remyelination is increased with aging in Tregs, and therefore they become like a problem for remyelination. And the other and probably more plausible hypothesis is that something that is required for remyelination is lost with aging, as most functions get impaired as we age, unfortunately. So we decided to test the second hypothesis and look at what is lost with aging that is needed for remyelination. But in order to do that, we needed to narrow down from 300 candidates to something that is plausible to test in the lab. And to do so, we went back to both our demyelinating lesions and our in vitro model. And we saw something that was quite interesting. Here is just an in vitro co-culture of OPCs with Tregs, and here is just a demyelinating area of one of our EAE models. And what you can see in both of them is that Tregs, which are identified in red here, or like by the expression of CD4 and FOXP3 in vivo, are always in close proximity to oligodendrocytes or OPCs, which are identified here by OLIG2, and in the case of in vitro by OLIG2 and N2. They are literally in close contact with each other in both models. So we wonder whether the effect that we have seen both in vivo and in vitro requires cell-to-cell contact, and this will help narrow down candidates and finding molecular mechanisms. So we did a very simple assay in vitro. We took our neonatal OPCs, which are primed to differentiate. We put them with young Tregs in co-culture, as we have done before, or we put them with young Tregs, but in a transwell, in a way that they will share their media, but they will not contact each other. And what we observed was that while the direct contact ones were enhancing oligodendrocyte differentiation and MVP expression, this was lost when Tregs were not in direct contact with OPCs, suggesting that at least some of the mechanisms might require contact, cell-to-cell contact in order to be efficient. So we next wanted to narrow down the candidates by using this information and the publicly available data sets. So what we did to try to narrow down those 300 candidates was to get those proteins that were expressed in the membrane in Tregs and were decreased with aging, and combine this with publicly available data sets that shows those genes that are enriched in OPCs versus oligos, and also were expressed in the plasma membrane. And then we did a protein-protein interaction analysis. We came across about 20 candidates, and then taking into account the number of binding partners and a few other factors that I'm not gonna detail here in the interest of time, but happy to discuss later on. We narrow down this to 7 candidates that we test in vitro. And out of these 7 candidates, 2 gave some interesting results. So we did a very simple experiment in vitro in order to try to see if any of these molecular mechanisms that we identified were involved in the regenerative capacity of Tregs. So what we did was we went back to our in vitro models and we took OPCs alone, OPCs with Tregs as our positive control. We know that young Tregs can drive OPC differentiation into oligodendrocytes. We have our control isotype, and then what we did was to block some of these receptors in the Tregs with an antibody before we put them in contact with OPCs. And what we observed was that when we were doing this with an isotype, we still saw an enhanced OPC differentiation, as you can see here by the expression of MVP. But when we blocked 2 molecules, MCAM, which stands for melanoma cell adhesion molecule, or IDGA2, which it's integrin alpha-2, we lost this pro-differentiation effect. When Tregs were not able, well, these 2 molecules were blocked in Tregs, and then they were not able to contact OPCs through whatever receptor they contact, which we have identified. OPCs could not have that accelerated differentiation into oligodendrocytes, suggesting that these 2 were 2 potential mechanisms by which Tregs enhance OPC differentiation into myelin-forming oligodendrocytes that were lost with aging. if that's the case, it's plausible that these two molecules could directly affect OPC differentiation, and maybe we could use those molecules as a direct effect in like OPC differentiation. So we tested that in vitro. We first exposed neonatal OPCs to MCAM in increasing dose, and we saw no effect. OPCs were differentiating in the same level as control. But when we did this with integrin alpha-2, we did see an enhanced differentiation of oligodendrocytes and also a change in the morphology into a more complex and branched oligodendrocytes, which is usually indicative of a more differentiated stage of the oligodendrocytes, suggesting that even if both molecules were associated to the Treg myelin regenerative capacity, only integrin alpha-2 can drive differentiation directly. The other one will potentially trigger another mechanism that we haven't identified yet. However, this doesn't fully prove our hypothesis, and if this is the case, and these two molecules are involved in the decreased aging capacity of aged Tregs to drive myelin repair, when we put Tregs in a young environment, they should recover this level. So that's what we wanted to test next. Unfortunately, this it's only a supplementary figure, and it's preliminary data because the N numbers were not that big, so it's not very robust yet. But what we did was we went back to the same in vivo model as before. We got the FOXP3 DTRs in which we depleted our endogenous Tregs, and then we took Tregs from young and aged mice, but this time expressing RFP, so that we could distinguish them by flow cytometry. And then we look at the expression level of these two factors, MCAM and integrin alpha-2 by flow cytometry. So what we saw here in both cases for MCAM and integrin alpha-2 is that with age, we have a decrease in the expression levels of these two molecules. However, when these two young or aged Tregs are injected back in a young environment, we didn't see an enhancement for MCAM in this case, but we did see that the levels of integrin alpha-2 are restored back to young levels, suggesting that indeed one of the things that happens when we put aged Tregs in a young environment that rejuvenates and contributes to myelin repair is that they get their integrin alpha-2 levels back to what they had in the young Tregs. So with this, I just want to go down to the conclusions, and hopefully we'll have some nice questions afterwards. So basically, one thing I wanted to establish in the seminar today, that I think it's quite well established now that the CNS is not immune privilege. CNS immune interactions are key for many functions in the CNS, learning, memory, but also for myelin repair. So T cells are not only bad for myelin, they are also needed to regenerate myelin properly. Aging affects not only the intrinsic capacity of the CNS to repair myelin, but also how Tregs can drive myelin repair in the CNS. With aging, Tregs have an intrinsically impaired regenerative capacity that it's highly relevant when we think about potential Treg-mediated therapies for myelin repair in MS. But fortunately, this process or this intrinsic impairment to regenerate myelin can be restored. They can be rejuvenated and brought back to their young Treg level capacity. So they are plastic and they can be manipulated in one direction or the other. And we have identified two mechanisms that are involved in this age impairment of Tregs. One is MCAM, which it's associated to OPC differentiation, but apparently not directly. It does trigger some other mechanism that we haven't identified yet. And the second one is integrin alpha-2 that appears to enhance OPC differentiation directly and is restored back to young levels in our like rejuvenated system. So this kind of opens quite some lines of questions, and obviously there's a lot of work to do afterwards to try to see if we could directly manipulate integrin alpha-2 or MCAM to enhance myelin repair in the HCNS, something to do still, but hopefully we are all working on. So with this, I would like to obviously first thank the whole amazing team that was involved in this like humongous project that started about 5 years ago. Denise Fitzgerald and myself that led the project, all the people in like Cambridge, Gdansk, here in Alicante, my previous supervisor in Altos Labs, who is now also involved, and all the people in Queens that contributed to this project, because this was one of the projects that survived 3 years of pandemic and moved to Alicante. So it was quite some work there. And obviously, I would also like to thank my current group here in Myelin Alicante Labs, the 2 other PIs that run the group together with me, all the people in the lab, collaborators at both institutions, and my funding. And I would like to thank you all for the invitation. And obviously, I'm happy to take any questions either in English or Spanish, whatever is easier.
Muchas gracias.
Bien, bueno, muchas gracias, Alerie, por la charla, excelente. Bueno, entonces queda abierto para preguntas, pueden ser en inglés o en español, ¿sí? Como prefieran y se sientan más cómodos. ¿Y Patricia?
Sí, Alerie, mil gracias por muy buena charla, muy interesante. Muchas cosas por avanzar en este campo. Y me pregunto si evaluaron cuál es el patrón de interleuquinas que secretan las Tregs en el joven y en el envejecido, para saber si eso también puede estar influyendo en la respuesta de la migración de los OPC y su diferenciación.
Sí, o sea, nosotros directamente no lo evaluamos, pero justo creo que el año anterior se publicó un paper en JCI que miraba algo parecido en el contexto de lung injury y influenza y la gripe, el virus de la gripe. Y sí que veían cambios en algunas citoquinas, tanto proinflamatorias como antiinflamatorias, y en nuestro transcriptoma se veía un aumento de las proteínas, perdón, de las citoquinas inflamatorias que no se expresan tanto en los jóvenes, pero sí en las viejas, y sí que podría estar influyendo. Yo siempre digo que al final creo que el campo de las Tregs ha intentado separar mucho su función antiinflamatoria de su función regenerativa, y creo que eso no es posible. En cuanto a si eso afecta a la migración de los OPCs, creemos que no, porque hay 2 datos que no he mencionado por interés del tiempo. En los jóvenes no vemos cambio en la migración de OPCs a la lesión, ni con las jóvenes ni con las viejas, pero hemos hecho el experimento inverso, en el que hemos cogido animales viejos y les hemos metido células Tregs jóvenes para ver si eso rescataba. Y en ese caso hay un impairment del recruitment que no se rescata con ninguna de las 2, pero sin embargo las jóvenes sí que aumentan la diferenciación. Entonces yo creo que el efecto es más directo en la diferenciación y no ni en la proliferación ni en la migración.
Buenísimo. ¿Y la microbiota de los viejos cómo está?
Fastidiada, no hay nada que el envejecimiento no arregle. En este caso no lo miré porque era otro proyecto que está ahora terminándose, pero sabemos que tienen de por sí una menor capacidad de fagocitar y un perfil más tipo M1 o proinflamatorio en el caso de los envejecidos, y las Tregs parece que aumenta su capacidad de fagocitosis en un cierto extent. Entonces, creo que no es correcto decir que el efecto de las Tregs es exclusivo en los OPCs, sino que tiene un efecto, digamos, multicelular que se traduce en un aumento de la diferenciación. Y sí que sabemos que también hay un efecto directo, que por eso tiene un efecto en cultivos, porque los cultivos son puros. Pero en vivo yo creo que es una combinación de varios factores.
Genial, gracias. Muchas gracias, Alerie.
Bien, ahí Ruth, ¿tiene una pregunta?
De ignorancia nomás. ¿Qué pasa con la barrera hematoencefálica en todo este proceso? ¿Está siempre disponible para que pasen las células o no? Esa es una pregunta. Y te hago una pregunta más cortita, así te la hago más corta. Con los OPCs que se diferencian, ¿se repueblan? ¿Con qué velocidad? O sea, pasan a ser maduros. ¿Y con qué velocidad se repueblan para tener más disponibilidad de OPCs? O recuperar
Vale.
el múltiplo de OPCs.
Empiezo por la primera. En el caso de la esclerosis múltiple, obviamente la barrera hematoencefálica está dañada, y en el modelo de lesión por toxina la dañamos nosotros porque al final entramos con una aguja. Entonces hacemos un modelo. Lo que pasa es que sí que
se mantiene, en los otros modelos en que trabajamos inflamatorios, la barrera no siempre permanece abierta. Hay un movimiento de integridad de la barrera.
Exacto, no se mantiene abierta todo el rato, pero sí que se abre, lo cual facilita la entrada. El Pero también en un artículo que trabajamos en colaboración con Adrian Liston en Cambridge, también vimos que no necesitan o sea, pueden atravesar mediante el mecanismo activo las T-Rex y llegar al CNS en un healthy system en el que no haya rotura de la barrera hematoencefálica. Lo que pasa es que obviamente la barrera hematoencefálica lo facilita. En este modelo creo que se cierra bastante pronto. O sea, nosotros la rompemos con la aguja al crear la lesión, pero a los 3-4 días está regenerada, al menos en el animal joven. No sabría decirte si en el viejo se regenera con la misma rapidez. Mi deducción es que no. Pero como no vemos cambios en la infiltración de T-Rex ni en jóvenes ni en viejos, creemos que no es uno de los factores limitantes. En cuanto a los OPCs, sí, los OPCs son células madre, por lo tanto tienen dos funciones. En cuanto tienen una diferenciación asimétrica, o sea, se dividen y generalmente una de ellas repruebla y la otra mantiene la población. No recuerdo ahora, on top of my head, esa división porque además cambia en la lesión versus el healthy tissue. Hay un artículo de William Richardson en el 2013 que hacen el etidio de bromuro y te dan todo el time course de proliferación, y creo que estamos hablando de días en un healthy tissue, pero no recuerdo el número exacto, ya lo siento, de las horas, y sé que en la lesión es mucho más rápido. En la lesión estamos hablando de 48 horas y in vitro, si los pones en ciertas condiciones, se pueden dividir cada 24 horas. Entonces depende mucho de los estímulos que tienen, pero sí, no hay... Hay un estudio de Robin Franklin del 2003 en el que ellos hicieron varias lesiones repetidas en el mismo punto para ver si había una depleción de los OPCs por exhaustión, de tener que diferenciarse, y no ocurre. Siempre mantienen la densidad, al menos en ese modelo. En esclerosis múltiple puede ser diferente porque pueden presentar antígenos y ser también targeteados en menor grado que los oligodendrocitos. Entonces puede haber una depleción activa por el sistema inmune. Pero por eso hay algunos pacientes que tienen menos OPCs, que puede ser por un fallo en el reclutamiento o porque haya un target inactivo, pero eso no se sabe aún cuál es el factor que regula si un paciente tiene OPCs que no se diferencian o menos OPCs.
Muchísimas gracias.
Gracias.
Bien. Jole, Dini.
Hola, ¿qué tal, Alerie? Felicitaciones, me encantó la charla. Una pregunta que quizás no tenga mucho que ver, pero ¿han visto o han evaluado el patrón o sabés si alguien lo ha hecho, el patrón de secreción de vesículas extracelulares por parte de los T-Reg? O sea, ¿tienen algún...?
Estoy segura que se ha evaluado en el contexto de inmunología, casi seguro que sí se ha evaluado. No sé si se ha evaluado el
Pero en
cambio
este contexto de animales jóvenes y viejos.
en jóvenes y viejos. No me atrevo a decirlo. Yo creo que no, pero se me ha podido escapar algún artículo muy inmunólogo que no esté tan centrado en la neuromología y que no lo tenga en mi radar. No, yo no lo recuerdo, pero es posible que se haya hecho. En el contexto del sistema nervioso central, seguro que no. Eso sí que no, porque nosotros las consideramos porque, bueno, en el artículo anterior de Nature Neuroscience lo hacían con secretoma y vimos varias formas de fraccionar el secretoma y tal, y uno de los candidatos era vesículas extracelulares, pero no llegamos a adentrarnos mucho. Ahí lo dejamos como es una posibilidad, pero no la miramos. Pero ahí la
Porque
verdad es que
lo
no lo sé.
descartaron por esto de que establecieron que tenía que ser necesario el contacto.
Exacto, aquí sí. A ver, yo creo que hay varios mecanismos. En este
Sí.
es un contacto, pero si por ejemplo las T-Rex están activadas, también es el secretoma. O sea, depende del estadio en el que estén las T-Rex, que lo que pasa es que ahí no he entrado porque ya empieza una inmunología más compleja. Yo creo que hay múltiples mecanismos. Hay un mecanismo de contacto y hay un mecanismo de secreción que creemos que podía ser vesículas extracelulares, pero identificaron en el artículo anterior la proteína CCN3 como uno de los factores. Y sé que estaban mirando algo de vesículas, pero nunca llegaron a adentrarse ahí, entonces no sabría si tienen un rol. Lo más probable es que sí, pero no lo sabemos.
Y otra pregunta, en estos animales viejos, no, jóvenes depletados a los cuales se les ponen las células de los viejos, ¿hay un cambio en el outcome motor? O sea, ¿ustedes evalúan el comportamiento motor o alguna alteración en este modelo de esclerosis?
En este caso
A nivel comportamiento.
no se evalúa el comportamiento motor porque la lesión es focal, es 1
Ah,
milímetro cúbico de lesión,
está bien.
porque es la
Bien.
única forma. Hacemos este modelo porque nos permite evaluar de una forma muy clara la regeneración específica de esa lesión focal. Por ejemplo, la EAE, que es una lesión multifocal, que está mediada por el sistema inmune, sí que tiene un efecto motor, y la cuprizona también, porque son multifocales, pero el problema es que nunca sabemos el tiempo en el que está la lesión, en qué fase regenerativa está la lesión y por lo tanto evaluarlo es más complejo. Este modelo es una lesión focal muy pequeñita que nos permite evaluar muy clara la respuesta regenerativa y por eso lo usamos, pero al ser 1 milímetro cúbico no tiene ninguna alteración motora.
Y en otros modelos no se ha probado esto que estás planteando en este, ¿no?
En el joven y viejo no con las T-Rex, se ha usado en otros contextos y sí que se ven alteraciones motoras y lo usan como un readout si mejora la remyelinización, el cambio motor. En el contexto de jóvenes y viejos de T-Rex nosotros no lo hicimos. En el paper anterior lo hicieron en cuprizona, pero no hicieron el testeado motor, lo hicieron también por histología.
Bueno, muchas gracias.
Gracias.
Ana.
Hola, sí, buenas tardes, Alerie, hermosa tu charla, aprendí mucho. Y decime, en el contexto del envejecimiento de los OPC, que se sabe que tanto por factores extrínsecos como intrínsecos los OPC se les altera el funcionamiento mitocondrial, la cadena respiratoria, la síntesis de ATP, las especies reactivas del oxígeno, el acortamiento de la telomerasa, en fin, te puedo citar todas las razones que llevan al envejecimiento del OPC. Mi pregunta, yo te escuchaba, me pareció sumamente interesante lo que planteas, pero digo, ¿cuánto pesa el envejecimiento del OPC que pueda hacer que el T-Rex no tenga efecto porque está predominando el envejecimiento del OPC, ¿no es cierto? Porque esto va a ayudar sin lugar a dudas, pero si vos tenés un individuo envejecido con OPCs que no tienen capacidad de diferenciación y maduración, siempre es el resultado de muchas cosas que pasan simultáneamente, ¿no? Y yo creo que todos los que trabajamos en esto queremos aportar un granito de arena para favorecer el sistema, pero me pregunto.
Al final es cierto que dices, el sistema no es algo lo que falla, fallan muchas cosas a la vez y hay una pregunta que siempre está de si el OPC puede escuchar, el OPC envejecido puede escuchar las señales que le manda el T-Rex. No lo he metido aquí por 2 razones, por tiempo y porque el experimento tiene unos caveats, sí que está en el paper. Hicimos el experimento inverso en el que a animales viejos le quitamos las T-Rex viejas y le metimos T-Rex jóvenes y viejas. Y las jóvenes sí que mejoran parcialmente la diferenciación, por lo que sí que hay una pequeña capacidad del OPC de escuchar. Si es de ahí a que sea capaz a rejuvenecer el sistema completo, como lo puede hacer una calorie restriction, no. Estamos hablando de un factor que, como bien dices, aporta un granito de arena. Pero al final, si tenemos en cuenta que los pacientes no tienen ni una sola terapia que aumente la remyelinización, supongo que cada poquito mejora, pero nos dio bastante alegría ver que cuando cogemos animales viejos, y estoy hablando de animales de 23, 24 meses, y les quitamos las T-Rex, les sometemos a la lesión, les metemos T-Rex jóvenes, las jóvenes aumentan significativamente los oligodendrocitos. El problema es que ahí es más interesante porque en los viejos hay un impedimento de recruitment cuando quitas las T-Rex que no se recupera del todo, pero sí que las jóvenes todavía tienen un pequeño efecto, no es una rejuvenecimiento completo para nada, y las viejas no. Entonces, y in vitro hicimos unos experimentos que no están en el paper con OPCs viejos y también aumenta la diferenciación un poquito in vitro cuando las pones con T-Rex. Lo que pasa es que no llegamos a tener una N robusta y por eso no lo pusimos en el paper. Entonces, parece que sí que tiene la capacidad de escuchar, por decirlo de una forma simple. Obviamente no es un rejuvenecimiento completo.
No, perfecto, pero hace algo, porque justamente diferenciación y reclutamiento se ven muy afectados, ¿no? En el OPC. Fantástico. Muchas gracias.
Gracias.
Bien. ¿Hay alguna otra pregunta? Yo tengo una. ¿Tenés candidatos o ideas de qué es lo del ambiente joven? ¿Qué molécula o qué señal hace, por ejemplo, que regule integrina alfa 2 en células viejas? ¿Hay algún candidato? ¿Tenés alguna idea o no? ¿O es algo...
No hemos mirado el abstract, si te soy sincera. Nos sorprendimos que pudiéramos ver como tan redonda el integrin alfa 2, ¿no? Que haga un efecto directo en los OPCs y lo rescatemos, pero no tenemos ni idea. No te podría dar ningún candidato porque estaría lanzando una bola al aire sin ningún tipo de backup. Sería interesante. Sí que es cierto que creo que lamentablemente de momento no está en las cartas. Siempre vamos más a la interacción T-Reg o PC, pero desde luego sería algo interesante de ver qué es lo que lo sube. O, por ejemplo, un experimento que no se lo había planteado nunca Robin, se me acaba de ocurrir, sería ir al calorie restriction, que hizo Robin en su momento en un paper en Cell Stem Cell que rejuvenecía prácticamente completamente la remyelinización y ver si esas T-Rex tienen más o menos. Al final, calorie restriction es algo sistémico, pero no tengo idea de ningún candidato.
Bien, perfecto. Y otra pregunta también, esto es de total ignorante en el tema, con respecto a las otras células T que ejercen una función opuesta a la Treg en el sentido de que por ahí inhiben la remielinización. ¿Esos también se han estudiado qué pasa con la edad? ¿Aumenta la función en la edad o no se ha estudiado?
Sí que se habrá estudiado en el contexto de la inmunología seguro y sé que se vuelven más proinflamatorias en el caso de las TH1. En el caso del contexto de la remyelinización, Denise Fitzgerald, que era mi mentora en Belfast y ahora ella sigue con el tema, sí que está mirando las funciones de diferentes tipos de T-cells en este contexto y te puedo decir que son sorprendentes, no todas son tan claras, tan blanco o negro. Y nosotros en este modelo hemos visto que aumenta, una vez quitas las T-Rex, aumenta la infiltración de las otras T-cells, pero aumenta igual en el joven y en el viejo. Entonces no sabemos si ahí puede cambiar la función. Seguramente, como todo lo asociado con el envejecimiento, haya más proinflamación y haya más más T-cells inflamatorias en una lesión vieja que joven. Lo hemos mirado en el contexto de AA y no hemos visto grandes diferencias, pero no a nivel funcional.
Perfecto. Bueno, muchas gracias. ¿Hay alguna pregunta más? No.
Una
Bueno.
solita puedo hacerte, Alerie.
Sí, claro,
Claro.
¿Tienes experiencia en BREG, en linfocitos B reguladores? Nada.
No, tengo que decir que mi parte de inmunología se limita a que sé que existen y sé que son claves en la progresión de la enfermedad porque casi todos los medicamentos asociados a B-cell depletion son los únicos que funcionan en la fase progresiva de la esclerosis múltiple. De ahí a qué papel tienen las BREG, no. Sí que te puedo decir que, siendo honestos, también estarían eliminadas en nuestro sistema porque expresan el FOXP3. Pero qué porcentaje contribuye, no lo sé. Hay un único paper que yo recuerde en desarrollo en el que quitan las B-cells y hay problemas en la mielinización en desarrollo, pero quitan todas las B-cells, no las B-regs. En remyelinización no lo he mirado en detalle, pero no lo he visto. Creo que la parte reg de las B se nos suele olvidar a todos los que no somos inmunólogos.
Muchas gracias.
Gracias a vosotros.
Bueno, entonces, si no hay más preguntas, damos por finalizada la conferencia. Otra vez, muchísimas gracias, Alerie, por la predisposición y por la charla. La verdad que fue fascinante.
Me alegro.
Eso y bueno, estamos en contacto. Sería bueno en algún momento tenerte acá en Argentina.
Yo encantada. Hace mucho que no bajo por Argentina, así que yo encantada estaría de ir en persona.
Buenísimo, buenísimo.
Bueno, muchísimas gracias.
Gracias
Bueno,
a vosotros. Buen fin de semana.
Gracias a todos. Chau, chau.
Chao, gracias.